Hi, everybody. Welcome to Love Goes Live. My name is Mark Lubkowitz. I'm a professor of molecular biology at St. Michael's College here in Burlington, Vermont. And I've been making videos on COVID-19 and other sciencey stuff for the St. Mike's community and really for anyone who's interested in science. And before I forget, if you're enjoying this content, please subscribe to the channel so I can stop spamming my friends with emails. So if you've seen other Love Goes Lives, you've heard me say that science is both incremental and self-correcting. And today's story is one about incremental progress, and that is getting closer to finding out the sequence events of how COVID-19 ended up circulating in humans. Okay, so without any further ado, let's get to the paper, shall we? And this paper was published in Nature. All right, let me get my PowerPoint presentation up and running here, as I always do. Okay, great. Okay, so at this point, it's pretty well established that COVID-19 is a coronavirus that came to humans from bats. It's about 96% identical, over 29,000 plus nucleotides to a coronavirus that's found in bats. It's also known that a small portion of COVID-19 came from pangolins. And actually, arguably, it's the most important part of the coronavirus because this is the part that enables it to actually get so efficiently into human cells. So what's not known, though, is did this virus go from bats into a pangolin? Or did it go from a pangolin into a bat? And when I first started looking at this and reading about it, I started asking the question, well, what are the probability that these two animals even ever come in contact with each other? Well, it turns out that they're both they occupy the same ecological niche. They're both insect feeders, they're both nocturnal, they're both found in the same habitats. And so at some low frequency, I don't think it's a high frequency, but at some low frequency, they come in contact with each other and they both happen to carry coronaviruses. Now, the million-dollar question, of course, is when did it jump, or from which species did it jump into humans? So the paper that I just got is an accelerated article preview. So this means that Nature, it hasn't been published yet in Nature, it's about to be published in Nature. Nature felt it was important enough for the scientific community to know about it that they put out a preview. And so I apologize for this, but it's been stamped with preview all across the paper, and so you'll see this over and over again. All right. So this story begins in 2019. So penguins are illegally trafficked. They are the most illegally trafficked mammal on the planet, interestingly enough. Not by mass. If you have one elephant, that's worth a lot of penguins. But as far as sheer numbers, they're the most largest number of illegally trafficked mammals are penguins. And so there was a sting operation in China. They seized a bunch of penguins and they took them to a wildlife rescue clinic where they all proceeded to die. And because they do carry coronaviruses, someone had the foresight to actually take these animals and put them into a -80 freezer so they would be preserved. And then fast forward into 2020, and this group of scientists went back to that freezer and they asked a very simple question. Did these penguins that died, did they have lung damage that would suggest a respiratory disorder? The answer was yes. And then they asked the question, can we recover a virus from them? And they in fact did. In fact, it was a coronavirus from a pangolin. All right. Let me show you some data. This data, it looks like a lot, but it's actually an unbelievably efficient visually way to scan through genomes. And here's how this works. So down here, these numbers, there's a nucleotide number. So this represents the entire coronavirus genome. So a little shorter than 30,000 nucleotides. And then we're looking at similarity scales here. And what they're doing is they have 3 different bat coronaviruses. And then this is COVID-19 in red. And then they're comparing it to the pangolin coronavirus. Here's how you want to read it. There's really only 2 things you want to see. You can see this almost right away, is you're looking for lines that line up on top of each other. So remember, this red line is going to be COVID-19. The green line is the bat coronavirus, which is pretty well established where it came from. And you can see this, right? So the red and the green lines are on top of each other. In fact, there's so much on top of each other, you can't even distinguish them. So red and green, right on top of each other. Red and green, right on top of each other. All right, and then all of a sudden you get to this point here where they're not on top of each other and they really start to break away from each other. And so the other thing you can see on these type of data displays is you can tell where recombination has occurred. And so everywhere that you see a line on top of it, that means that these viruses are very similar. And when they dip down like this and the lines separate from each other, that suggests that there's been a recombination event. Okay, so what this tells you is that COVID-19, the majority of it, remember that's the red line, is very similar to this green line, which is that bat coronavirus. But there's this region in here where it doesn't seem to look like that bat coronavirus. Once again, this is sort of old news. This was established by an earlier group. So let me go back a slide here. So we're just going to now zoom in just on this region right here. So let's zoom in this one region which doesn't look like bat RNA. All right. So if you zoom in, coming into it, remember red is COVID-19, green is our bat coronavirus. They're very similar to each other, and you can actually even see that here in a phylogenetic tree. Here's COVID-19, here's its closest relative, it's a bat coronavirus. Then when you zoom into this region right here, remember red is COVID-19, it doesn't look like that bat coronavirus anymore. And so if you now compare it to that recently identified pangolin coronavirus, lo and behold, there's COVID-19. And for this region, its closest relative is that pangolin coronavirus. So what is this region? So if this part came from pangolins, what is it? So if you've seen other Lubbock's lives, we know that this is where the spike protein is. The spike protein is what binds to the ACE2 receptor. This is how the virus gets into human cells. So what this is telling us is the part of COVID-19 that is necessary for getting into human cells came from a pangolin. And then just to finish it out, this region here, so you can see the red and green are back on top of each other. Here's COVID-19, its closest relative is once again the bat. So take a message, you got bat, a little piece of pangolin coronavirus, more bat coronavirus. That is what led to the creation of, or the evolution, I should say, of COVID-19. So what is recombination? I've read about it in papers, I've bantered it around today, and I have yet to explain it. So let's actually look at what recombination is. And the recombination is just what it sounds like. It's when genetic material recombines. There's a bunch of different mechanisms. The way it happens in the viral RNA world is really interesting and shockingly frequent. I was reading about this morning through a bunch of review articles, and I was like, wow, like who would have thought this happened so often? And so when you have two different viruses in the same cell, so you have a red virus, you have a blue virus, they're RNA viruses, they get copied by replication machinery. They're called RNA polymerases. You and I, they'd be transcriptional machinery for those of you who are checking my vocabulary usage. So in viruses, though, we're going to replicate this with this machinery. And here's what you want to remember. It's not very good at its job. It falls off what it's trying to copy very frequently. And in my world, that's called processivity, and which is a mouthful. And so when you're copying machinery and it falls off, it can pick up on another template. And I was trying to think of an analogy, and actually it happened to me this morning. I was copying over some notes. My kids interrupted me. And when I started copying over again, I picked up at a different place. And lo and behold, I ended up with a chimeric note page, right? And so that's essentially what we see here. We've got a red virus, we've got a blue virus. When it falls off and it starts copying, you end up with this chimeric. So let's unpackage that a little bit more, because I know that was a little bit complicated. So let's say we have this virus that's red. We have some machinery that's copying it. It falls off because it's just not very good at what it does. There's another virus in the cell. It gloms onto that virus. That template starts copying it. And now the resulting products, this is going to be a novel virus, is chimeric. So it's half of the red and half of the blue. So here's what they think happened in bat, pangolin, human story. So you've got a bat coronavirus. It starts being replicated inside of a cell. It's chugging along, replicating, copying, everything's as it should be from the coronavirus's perspective, and then it falls off of its template. So now here you have that machinery that's copying the virus. It's got this product that's only partially copied, and there's yet a second coronavirus in this cell. This one happens to be from a pangolin, and what does it do? It attaches and starts replicating and copying that. It falls off again. So now here we have the machinery that's doing the copying, and we've got a piece of RNA that's from a pangolin, a piece that's from bats, and lo and behold, it reattaches to that original bat coronavirus and finishes copying. And now we've got something that's 96% identical to the original bat piece with a small chunk that's from pangolins. All right, so let's put this back in biological context, and particularly in clinical context. So when you think about viruses that are jumping around between species, very often you have a reservoir. So the reservoir would be a species that is not affected by the virus, but is carrying it. At some frequency, usually at a very low frequency, that virus will jump to an intermediate host. If that intermediate host happens to have if it happens to carry viruses that are typically in the same family, you will get recombination. And at some frequency, that host will get sick. And then at a low frequency, that new novel virus can get transferred to other animals, including humans. Now, why was this paper so profound? Remember that their first clue was they found pangolins that had died of a coronavirus. The reservoir, if you remember, rarely gets affected by the virus that's carrying, but very often the intermediate host is the one that gets affected. The fact that these pangolins died of this coronavirus suggests that they were the intermediate host which puts the reservoir as bats. So there's stories suggesting that bats gave the coronavirus to pangolins. There was a recombination event. And now the question is, did it jump from there into humans? Let me zoom out here for a second so I can see everybody. Okay, so I think here's the take-home message. So remember that bats and pangolins occupy the same ecological niche. They're both insect hunters, they're both nocturnal, they both carry coronaviruses. At some frequency, and let's be honest about this, it's low frequency. At some frequency, they will come in contact, or the viruses will come in contact, and you'll get some recombination event. That ends up, let's say, in a pangolin. And then the question is, what's the probability that it's going to enter into humans from there? Well, here's where that most illegally trafficked mammal comes into the story. So the probability of a coronavirus jumping from a pangolin into a human is a low probability event. But if it's the most trafficked animal, illegally trafficked animal that humans traffic, those low probability events become much higher. That's the take-home message. Okay, that's all I got for you today. My name is Mark Lubkowitz, trying to let crisis bring out the best in me. And as always, rising from the ashes like the phoenix. That's all I have to say from St. Michael's College. Actually, from my living room, pseudo St. Michael's College. See you next time. Bye.